got Dr. Dario Chavez with UGA. We're going to talk about fertility management.
Good afternoon, everybody. Today, I am going to present a bit of the work that we have been doing since I started in 2014. When I started, I wanted to set up a long-term project where we can follow the nutritional status, both in soil, leaf, and fruit of an orchard, and try to set up something that we can start looking at the guidelines that we normally are using right now in Georgia and southeastern U.S. And the main idea that I have is basically working with just nitrogen, because we thought it's one of the nutrients that is widely used, and also some of the use of the nitrogen is a lot of the spends that you have in an orchard. So if we can optimize the fertilization management for these nutrients, it will be helpful for peach production. So more than telling you what I think so that will be the best thing to do, what I want to do is also kind of create the opportunity to talk about it, because I'm sure you guys have nutrient analysis of your orchards that you do year after year, and maybe I just want to bring the attention of things that I am seeing in these plots, and maybe something that will be in common with what you are seeing, and maybe we can come up with better recommendations and solutions for the future. So just talking about the previous research that we have been doing from 2015 to 2019, and this is something that we have already presented previously, but I just wanted to bring the points really across. Is what we did is investigated the effect of irrigated versus non-irrigated trees, and then also looking at different irrigation system deployment, drip versus a sprinkler. But also we were looking at different fertilizer levels, and these levels were based on the recommended rate that you find in the Southeastern guide. So we were looking at treatments of 25%, 50, 100, and double that amount. And the idea was to look into using a highly productive cultivar, July Prince, that pretty much the standard of the industry for yield, and using Guardian as well. So what we found in that research is that irrigation since establishment for sure helped the trees. We had actually bigger trees, and also we had higher yield when they started producing fruit. And this was already tested again in another experiment using MP29 rootstock as well. And we found the same results, where basically irrigation does help the trees in our conditions, because in the southeastern U.S., although we think, we have rainfall often, we have periods of time that where we don't have the rainfall. And Dr. Pam Knotts yesterday was talking about that happening in Georgia, and how often it has been the last 10, 20 years. Another thing, drip irrigation, and this is what normally everybody knows, is more efficient. One thing that we found, and I will talk a little bit about this in the case of the nutrients, they are actually using drip irrigation. It makes the tree more efficient also for nutrient uptake. And you actually have higher concentrations of certain nutrients, and inclusive micronutrients, which are important for peaches as well. And the main point that we found is the higher fertilizer does not translate in a higher nutrient concentration in plants, and larger plant size or yields. And what we did is we spent several years basically trying to look at all the inputs and outputs in the case of nutrients, and to the point that we were measuring basically collecting all the leaves from trees, and then looking at the amount of leaves that we had, and then the concentration, quantity of nutrients, the same thing for fruit and things like that. And we found that basically when you compare all the nutrients across the board and all the treatments, we didn't see any difference, including the 25% fertilizer rate. Of course, when I finished this experiment, I thought 25%, it may be too extreme to be calling it. So we wanted to continue with the experiment, and we decided to drop the 25 and the 200%, and basically follow with the experiment of just the 50 and 100% of the rate. And then we basically started doing that. So the research from 2020 to 2022 was basically based on that. We decided that we needed an optimal fertilization for peaches, and also, the majority of the studies that have been used for fertilization management, I think so we come from different soil types, California, and also Mediterranean regions, different environmental regions. So we don't need a Georgia-specific, Southeastern-specific recommendations. Now, what we did, we had some field evaluations both in young and mature orchards. The idea was to be able to see what is the difference, physiologically speaking, that tree has in the different stages of life. And I wanted to go through a little table about the macro and micronutrients that we have in basically in plants. The ones that I wanted to focus on are calcium, magnesium. And calcium in apples is well known. It creates issues with bitter pit, and foliar applications of calcium are normally done in apples. And then magnesium, very important for chlorophyll photosynthesis. So that's an important micronutrient as well. In the case of the micro, we have boron, fruit set, pollination, very important. And then I want to focus on zinc and iron. Both of them are somewhat related to enzyme physiological process related to chlorophyll photosynthesis as well. So very important in the plant. So why I'm talking about those specific nutrients is because of the story that I'm going to try to tell. So I really haven't, finally I decided to go ahead and start mining a little bit of the data that we have from year after year. And we started looking at the nutritional status of the orchard from 2015 to 2019. And this, again, was July Prince and Guardian. We had some different treatments, irrigated versus non-irrigated, and then drip versus microsprinkler and the different rates, okay? And the idea was to be able to evaluate how the nutrients did in this. So this is a little bit the same way it's going to be presented in all the data. The difference will be what kind of nutrient we're talking about. In this case, it's micronutrients, and also the treatments. So this case is irrigated versus non-irrigated. Then you have the years, 17, 18, 19, and in here, this is the soil, leaf, and fruit. Now, the only things that I'm going to bring up are things that jump out of the story. So for example, I started noticing that calcium started really being high in the soil, and it started accumulating. And this is something that I will bring up in the next story as well. Now, in the leaf, we actually didn't have that effect, meaning that the plant was not able to uptake that calcium, and it was not moving in the plant easily. And in the case of the fruit and in the leaves, we had some deficiencies. So the deficiency will be presented by the arrow. So you can see that calcium and magnesium, in this case, we're having some deficiency in leaves, and in the fruit, we're coming as a deficient. So there was a disconnect from the soil to the plant and to the fruit, right? So that's a point that I wanted to make in there. In the case of irrigated versus non-irrigated, as I mentioned before, some of the nutrients do better with irrigation, and some others don't do that. But either way, that is one of the things that I wanted to bring up in here. Now, this is the graph of micronutrients, and the same thing, irrigated versus non-irrigated. We have in here the boron, zinc, manganese, iron, and copper. And you can see that in some cases, the non-irrigated level has a higher rate, basically because it's getting accumulated on the soil. The water is not, there's not a solution where the plant can take up those nutrients. And you can see that basically the same happens in here. Now, concentration of some of those compounds is going down in the soil and in the leaves as well. And some of those also are deficient. And the ones that are coming as deficient are iron and zinc. And they are coming in the leaf and in the fruit as well. So this is something that we're starting to look at here. Now, when we start looking at macronutrient distribution, looking at drip versus microsprinkler, remember that I mentioned that the drip is more efficient for water savings. But also, we found that in some cases, when we have the drip, some of the drip actually increases the concentration of some of the nutrients. And it will be more clear when you look at the data of this same orchard the next years. So, we have the same orchard. In this case, we're just presenting 3 years, but I will present the other 3 years in the next part of the presentation. Now, the same thing, magnesium, calcium, the deficiency in the leaves and the plant, and the fruit, sorry. And then when we look at micronutrients, same issue. You have iron and zinc coming up as a deficiency. Now, again, drip, those become more important in the amount of some of the micronutrients that are coming up in the plant, as I mentioned before, which was surprising to me looking at that in comparison with the microsprinkler. Now, when you look at fertilizer effects, and again, here we have 4 bars where we have 25, 50, 100, and 200%. And you can see in the soil level how it follows a gradient, because basically we're doubling. We have 25, 50, 100, and so the concentration of some of those compounds actually increases when you look at the treatments, right? So it follows that, and some of the other elements actually goes in the other direction because they get taken up by other elements as well. Now, calcium again is pretty high in there, and we're seeing that in this case, magnesium and calcium, again, is coming up as a deficient. And you can see that in the soil, the level of the magnesium is actually going down even when we're adding more, meaning that more likely calcium or magnesium are having some conflict where because we have more calcium, it may be binding and that making it that the plant cannot take that magnesium, and it may be a problem. Now, when we look at micronutrients and the different levels, you can see that it follows the same gradient, right, in the soil. Now, in the leaves, some of this maintains, but then you have the iron and iron and zinc, again, deficient. And you can see that the rate goes down with time. So what we are looking at here is the irrigation did enhance the nutrient uptake and nutritional content in leaves. And this actually corresponds to the data that we had where we had a bigger plant size and higher yield in some of them. And now the drip irrigation did have a better concentration for certain nutrients, specifically the micronutrients, which compared with the microsprinkler was interesting. Now, again, higher fertilizer rates did not really resulted in a higher nutrient uptake on concentrations. Again, we think that we can reduce at least half the recommendations currently available and basically work with that. Now, something that consistently comes along in all the analysis is the calcium and magnesium as macronutrients and zinc and iron as the micronutrients that coming up deficient, basically in the plant and of course in the fruit is not common. And this is something that I think so that it probably will be important to start looking into it. Can we do some foliar application of some of the nutrients or also soil application of certain nutrients that maybe, and also think about why these nutrients are being bound in the ground, basically. This may be important. Are you evaluating your water that you're using for irrigation? Some waters, if they have high carbonates, they can form bicarbonates and bind that calcium and magnesium in there, and it can create issues. And then you will need to change your type of lime that you're using. So those are things that are important because you're incrementing in soil, and you say, well, why is it not going to the plant? Now, if you think about spraying some of these micronutrients and macronutrients, some of them may not get into the pitch. So that is something that we probably need to start looking into the different formulations that we can find in the market, and if they would make any difference. Because, we don't know if this is something that's going on in all your farms, and that's why I wanted to bring it up, and maybe you already are addressing this, and it would be nice to talk about it. Now, when we look at the 2020 to 2022, looking at the same orchard, where we just started working with 50 and 100% fertilization rate, and the same thing, now we basically knocked out the non-irrigated trial and just started looking at a smart irrigation app that we have out, and a sensor-based app for irrigation. And then we have the drip versus micro sprinkler and just the 50 and 100. The graphs are similar, where we have micronutrients, we have a soil, leaf, and fruit. And then this is the same location where if you look the previous data, we had it from 15 to 18. And then the 19, we make sure to put the protocol in place and evaluate if we didn't have any carry over from the previous treatments that we were doing. And we make sure that there was not the case, and so then on, we jump into the next experiment. So finally, you can see that calcium still keeps building up in the soil. And then when you look at calcium in the leaf, finally starts appearing a little bit more in here. What I think that happened is that we had evaluated the irrigation water that we're having. So we have two sources of irrigation in our orchard. One that is ground from basically a water pump, and it has high bicarbonate. And the other is city water. And the pump broke these years. And basically we started using some of the other water. And we started having some better results with calcium. So I'm not sure if that's the response, what we got from that. But I was surprised that, however, in the fruit, still we don't see that effect, even when the plant is having a better look. In the case of irrigation, app versus sensor, we don't see differences between the two. Basically, once you have irrigation, the nutrient uptake concentration is pretty good. In the case of drip and micro sprinkler, remember that we mentioned that the drip gives you higher concentrations of certain micronutrients and macronutrients as well. You start seeing that in potassium, and also it flips a little bit in calcium and magnesium. And then you have the deficiency again appearing in the fruit, no matter what. In the case of the micronutrients, when you look at boron and zinc, we were having a good effect on the concentrations. And now the drip, you can see that it's higher for both elements, which it seemed something interesting about the drip effect. Now, when you look at the 50 versus 100% fertilization, one of the things that is important in here is to see that there's really not that difference between the nutrients uptakes for all of them in the soil. This is the soil level, but the calcium is still high. But in the uptake and the leaf, nitrogen is the one that's going to start differentiating because it has a higher difference rate of nitrogen. But then overall, in the fruit, still calcium and magnesium comes up as deficient. When you look at micronutrients, you start seeing basically that boron is in a good setup in the fruit, and also in the other elements, there's no deficiency. But then zinc and iron finally are staying in a good level. And these zinc and iron are basically in the age of deficiency in the fruit. So in this case, it changed a little bit that. Now, when we're looking at app versus sensors, we didn't see differences between each other in nutrient profiling. But we did see that the drip irrigation system actually helped getting a higher nutrient concentration. Now, I do think that using half of the rate that we are using, it is recommended. The nice thing about it is that if you start looking at deficiency of nitrogen, you can act up on it. and you are always collecting leaf samples in that year, so it's something that you can work on it. The other nutrients, you should not have a problem. Now, the deficiency in the fruit of calcium and magnesium, we always talk about calcium like the cell wall, fruit firmness and quality. So it may be something important to think about it, and also that may be affecting in some way or another. Now, finally, looking at a young orchard and similar thing, this is just a quick overview of what we think that may have differences as well in the future to think about. And here we have two different rootstocks, Guardian and MP29, and we had 3 different treatments of irrigation: up, sensor, and control, no irrigation, and the recommended rate of basically just 50% of rate of the rate. We didn't do 100% anymore. And what we decided to do is the same kind of setup. We have soil, leaf, and fruit. And one thing that started coming up often is that there was a difference between Guardian and MP29 for nutrient concentration that more likely is due to the anatomically differences between basically both of the rootstocks, which may be important for management later on, but it's something that maybe needs to be evaluated if we have more rootstocks available. Then another thing too is some of those levels change depending on what nutrient you are talking about. So it was interesting to see that sometimes and often Guardian had higher rates for different of the nutrients in comparison with MP29. Now, again, in this different study, we saw the same deficiencies, magnesium and calcium. And the calcium level in the soil was pretty high as well. And this is a different site. Micronutrients, when you look at iron and manganese in this case, are coming as deficient. So that's something that iron keeps popping up in there. And I'm not sure if it's important as well as we think in the case of the peach production. And maybe it's something that we haven't looked into it, and it may be important to do. And finally, when you look at the different treatments for irrigation, you can see that in the soil and leaf and fruit, you don't see a lot of differences, except that the irrigated treatments tend to have a little more concentration for micronutrients in the next slide. But again, magnesium and calcium keeps popping up as deficient in here. And then you can see that some of the micronutrients have higher concentration based on the treatments of irrigation. So this is a lot of information. And again, I thought the idea was to try to pass it along to you guys, because you are dealing with this information every day, basically, in your orchards. You have a lot of data that you have available, basically foliar analysis, soil analysis that you have been doing for decades. It may be something important just looking into the data that you have and maybe seeing that if you're seeing something that may be related to yield, plant health, and you know, I know that Juan Carlos, for example, has been doing a lot of studies with this skin bronzing related to nutrient, and maybe that's something that is affected as well from some of these things. So overall, I think so irrigation always important. It does help the nutrient uptake and also concentration in your plants. What it means that you need some type of water to be able to be moving in the tree. And then irrigation scheduling didn't really make a difference between the different nutrient profiles. And take home, always calcium and magnesium, that was coming up as a deficient in general across all the studies. And I will be curious to see if you are seeing the same things in your analysis, which may be something important to look. So finally, I think so drip irrigation is very efficient. We can work with the half of the rate of fertilizer that we currently are recommending. I think so some of growers are already doing it, either because of how higher the prices are right now. And you know, I think so that may be an opportunity to have your your rate. And also, some of the deficiencies will be important to look into it for foliar sprays or other types of things. I want to thank the Georgia Agricultural Peach Commission, the Georgia Peach Council, the block grant, Hatch grant, and also the multi-state SCRI. And this is my email, my lab site, and also the blog, and my personal cell phone if you want to chat and maybe you say that all the things that I say are wrong and you're not seeing that, I will appreciate that call too. So thank you so much, and if you have any questions.
Going back to the slide at the beginning where you showed what the pounds per acre things were in the second and third years. And then also, I'm sorry if I missed this, but did you split those up into multiple applications, or was that all?
Yeah, so what I did is I followed the recommendations from the guideline that I normally do. So the applications, it changed depending on the age of the tree. So the first years is 3 applications, and then after the trees mature is 2 applications, and basically 1 after bloom and 1 after fruit harvest.
Okay. Can you go to that slide where you showed your grade?
Let me see if I can find it faster. And this is the rate that is in the guide as well. And that's a 100% recommendation. Any additional questions?
Thank you, Dr. Calves. Next up, we have Dr. Elizabeth Sanowitz from Clemson University discussing peachtree viruses.
Okay, hi everyone. Thank you for having me here today to speak about our virology program. So we have a full hour dedicated to discussing viruses and virus diseases. So I'm going to cover 4 main topics today. I'm going to start off with results that are sort of hot off the press and we're still kind of finalizing, but that's our peach virus survey from South Carolina and Georgia peach orchards. Second, I'm going to give an update of the Southeastern Budwood Program from 2023. I'm going to give a brief update of the Clemson Clean Plant Center, what we've got going on, and then I'll also complete my talk by discussing our plans for upcoming research and activities at the Clemson Clean Plant Center. So we're going to start off with this virus survey results. So we did this survey using high throughput sequencing, and this is a relatively newer technology that has a few advantages for large-scale surveys like this. So instead of having to test every plant individually for every single virus, as you can see, there's quite a list of viruses that are known to infect stone fruits. Not all of these cause problems in peach. I will start right off by saying that, but some of them do. And this technology has the advantage of being able to detect not only the viruses that we already know about, but it can find all of those, and it can also find novel viruses, so viruses that we don't know exist yet, and then all virus variants as well. So even for the ones that we already know about, sometimes diagnostic tests are complicated by the fact that we could be missing certain variants or new variants of a virus. And so this technology, high throughput sequencing, has the advantage of being able to detect all of those. It comes at a higher price point. It's about $500 to do the high throughput sequencing on a single sample. So this is why it's not routinely used for diagnostics just yet. So the survey that we did that was funded by the Georgia Peach Council and the South Carolina Peach Council, this was in collaboration with Dr. Brandon's group at the University of Georgia. So you can see the sites that were sampled here on the map in Georgia in blue, and then in South Carolina in red. Dr. Brandon's group did the sampling in Georgia, and my group did the sampling in South Carolina. And the way that we did this was we worked with the growers to identify specific orchards or specific sites that were underperforming in some way, or they weren't performing in the way that they should be. And so we collected 20 trees, samples from 20 trees at each site. We pooled those samples, but we also stored tissue from individual trees in our freezer in case we wanted to go back to those individual samples later on. But we pooled samples of 20 trees into a single pool, and we had 20 of those from each state. So a total of 800 trees from around the 2 states were sampled for this survey. Before I get into what we found, I just wanted to tell you sort of what we were expecting and to introduce you to some of these viruses if you're not familiar with them. So viruses and viroids that we expected to find based on previous surveys and knowing what has been around for the past couple of decades, at least, are 2 of those viruses are called Prunedorf virus and Prunus necrotic ring spot virus. And these are closely related. They're in the same subgroup. And Prunedorf virus is known to cause stunting and shortened internodes, like you can see here in the photo on the right side. And then Prunus necrotic ring spot virus can be associated, it is a little different in different cultivars, but it can be associated with just kind of general thinning in the tree and decline over time. And some cultivars show like a reduced trunk circumference as well. Both of these viruses are transmitted in several ways, most efficiently through grafting. Also, they are transmitted in orchards via pollen, so one tree to another via pollen, and they are both also seed transmitted. A viroid, so the difference between a virus and a viroid is basically just that a viroid has its clothes off. A viroid is just a naked genome that doesn't have a protein capsid structure like viruses do. And they're very small, so up here you can see this is just the genome. It's a map of the genome and how it kind of folds back on itself and forms this odd secondary structure type thing. And although these pathogens are tiny, they can be mighty. They can cause some really important plant diseases. And these are some photos that you can see of the impacts of peach latent mosaic viroid. And we have been finding this viroid around the southeast now for a few years now that we've started really testing for it. So before I present the results, I want to have a couple of disclaimers about high throughput sequencing. First is that, as I mentioned, HTS detects all viruses. That is powerful and wonderful, but it's also kind of dangerous because we are able to see everything that's in those plants. That doesn't necessarily mean that all of those virus infections are relevant to growers or that they're causing any problems. Something that we teach in plant pathology classes all the time is that disease is the exception. And even though the viruses or the viroids might be there, a lot of times they are not actually causing problems. HTS is incredibly sensitive, so we can detect viruses at low levels in plants, and in some ways it might be too sensitive. In other words, it can be kind of prone to contamination, and it's really difficult sometimes if you have low levels of a virus that you're detecting to determine if that's a result of contamination or if it's a real infection. And so, it kind of opens up the door to follow-up studies. I will say at this stage, we can't associate any of the viruses that we find or will find with specific symptoms. That requires follow-up surveys. So I'm going to show first for Georgia and then for South Carolina what we found. And full disclosure, I'm going to show you everything that we found in table format. It will look like a lot of viruses, but I don't want you to panic. I think we can discuss what is important, and maybe that can be part of the discussion after my talk as well. Okay, so these are the results from Georgia. You can see we had a variety of different cultivars, so I'll just orient you to this table first. So we had a lot of the sites were collected from around Byron, and yeah, you can take photos of this if you want, and several different cultivars. And we found, so here's the column of whether we found peach slate mosaic viroid or not. And we've separated the virus detections into high levels of virus and lower levels of virus. We can discuss what that means maybe a little bit later. And I've highlighted in red or this orange color viruses that I think are important and that we should be managing for. So in Georgia, there were no real surprises. We found Prunus necrotic ring spot virus and Prundorf virus. We found peach slate mosaic viroid at a few sites. This is NSPAV is called nectarine stem pitting virus. And this is a virus that was discovered, I think, in 2017 on some material that was imported into Washington State. And they planted that variety in a germplasm block, and they found that those trees were showing signs of decline, and they detected this novel virus by high throughput sequencing in those trees, and they tentatively associated that with stem pitting symptoms. I forget exactly what the nectarine cultivar was, but that virus is detected a lot in high throughput sequencing data sets, and we don't know quite yet how associated that is with specific symptoms. It's also detected a lot in asymptomatic infections. These other viruses in the low levels column, I don't worry about any of them at this stage. This NEVM seems to be all over the place. This is nectarine virus M, again, another virus that was discovered in a high throughput sequencing data set, not associated with symptoms whatsoever. And we did discover a couple of novel viruses here. So this is a different analysis pipeline of the data, and you can see here the tabamovirus and sobamovirus. Tabamoviruses haven't really been discovered in fruit trees or in fruit crops in general. The best known tabamovirus is tobacco mosaic virus, and those are easily mechanically transmitted. We have no data so far on whether that could be important at all. So that will be the subject of some follow-up studies. So the analysis for the Georgia samples is completed, and let's move on to the South Carolina. Okay, I told you it was going to look like a lot of viruses. So same deal here with the South Carolina data. The only difference is that we have not yet had a chance to complete the analysis for the novel viruses. We'll get to that when we get back. So we found peach slate mosaic viroid almost everywhere. We found PNRSV also almost everywhere. PDV, Prundorf virus, was also detected. The other two viruses that I want to discuss here, apple chlorotic leaf spot virus, or ACLSV, in prunus has been known to cause kind of mottling of the leaves, bark splitting, and some leaf deformation in various peach cultivars. But most infections are symptomless. So again, at this stage, we don't know exactly what symptoms this virus is associated with. Cherry green ring mottle virus is symptomless in most peach cultivars based on what I could find in the literature. Again, requires some follow-ups. All of these other viruses have not been associated with any disease symptoms, and they are commonly detected in HTS data sets around the world. So I don't really worry about them either. It is a lot of PNRSV in South Carolina, right? If you compare this to what we observed in Georgia. So I mentioned we collaborated with Dr. Phil Brandon on this project, and the first thing I did when I noticed the big difference was I asked him about how they sampled. And there were some slight differences in how they sampled in Georgia and how we sampled in South Carolina that could explain this big difference. When we went to a block in South Carolina, we were specifically sampling from trees that had some sort of decline symptoms. And in Georgia, they worked with the growers to identify the blocks, and then they just sampled down a row or down a couple of rows. So we were targeting declining trees a little bit more in South Carolina, and that could partially explain why we found more PNRSV. Dr. Brannon's explanation was that Georgia peaches are better than South Carolina peaches, but I don't buy that. And I think it's probably mostly due to sampling differences. Another question that I had was whether our diagnostic tests are optimal. I will discuss in a few minutes how we have changed our diagnostic methods for the Budwood program recently. Because those of you who are familiar with the Southeastern Budwood program know that Prunus necrotic ring spot virus is one of those viruses that we test for. So you might be asking, why is it everywhere if it's included in the Budwood program? And I had the same question. And I do have a few explanations. Maybe we can get to that in the discussion. But we did double check that we looked at the sequences that we found of Prunus necrotic ring spot virus in the high throughput sequencing data sets, and we compared that to the test that we are now using for PNRSV detection, and we're good. So Elise did that analysis just the other day, and she came to my office and said, we're good on the PNRSV test, so we are good. We know that our test is going to be capturing all of the variants that, or at least almost all of the variants that are out there. So I also know that when I look at the list of varieties, most of these are included in the Budwood program, but not all of them. And so, we might be missing some varieties here as well. Okay, so this is just another view of the data showing that there are a lot of, keep in mind these were pooled samples, so we know that these could be co-infections in single trees, or it just could be individual viruses in those trees, but we analyzed pooled samples. And overall, seems that we found more viruses in South Carolina orchards, but again, I don't buy Dr. Brannon's explanation on that. So these are my overall conclusions initially from this survey. In Georgia, PLMVD was the most common, the viroid was the most common pathogen that we found, and then PNRSV and PDV at a few sites. We did find that novel Tabamo virus as well. At this point, it's unclear if it's going to be a problem, but we will do some follow-up studies on that this year. In South Carolina, we found that Prunus necrotic ring spot virus and peach late mosaic viroid were very common across these data sets. Numerous viruses that are likely not associated with disease were also detected. So again, it's kind of a double-edged sword with high throughput sequencing as a method. So for the novel viruses that were found or that could be found in the South Carolina data set, we will follow up on those this year. And I also want to say I took the farm names out of the tables that I presented, but of course if you're interested in the results from your specific farm, if yours was one of the places that we sampled from, I can pull up the data later, and we will also send out individual reports once we have all of the data analysis completed. I will say overall, I think this is good news. There were no major surprises, and we, at this point, again, something we can discuss after my talk is done, we're not recommending adding any viruses to the Southeastern Budwood program testing. I think we need to do some follow-up studies before we add anything. And I want to stress that this survey is a starting point for us. And some of the most immediate upcoming research to follow from this is that we will need to complete the novel virus analysis for South Carolina. We will follow up on potential symptomatology associated with apple chlorotic leaf spot virus. That's one candidate that we might want to think of adding to the Budwood program testing, but I think we have to do some studies first. And then we need to follow up on these novel viruses to determine if they're going to be a concern. Okay, so let's move on to the Southeastern Budwood program testing from 2023. So if you're not familiar with this program, it was started by my predecessor, Dr. Scott, in collaboration with the growers in South Carolina and Georgia and the nurseries in Tennessee. And this is a really excellent program that has, it was started in around, I think, the year 2000 or 2001, and it's still going strong. So I started at Clemson in 2019, and we've made some changes to the program in terms of how we're doing the testing. But it's a very effective, really good program. So we have The way this works is we set up blocks of 50 to 100 trees of each cultivar on farms in South Carolina and Georgia. And we collect leaf samples in the spring, and we test for, traditionally it was Prunedorf virus, Prunus necrotic ring spot virus, and plumpox virus. But now, as of 2023, we have added peach latent mosaic viroid to that testing regime. When we get the results from the testing, we produce maps of the block. So this is an example of a budwood block map, and it notes if there are any infected trees. So like green is Prunedorf virus and yellow is Prunus necrotic ring spot virus. So then we tell the nurseries that they should stay away from those trees. Because by far the most efficient method of virus dissemination is through grafting. And so if we can avoid that from the beginning, then you're at least starting off on the right foot. So these are some summary statistics from the 2023 program. We had 37 peach cultivars that were tested for the 4 pathogens. We had 2,343 trees that were tested, and overall low incidence of these pathogens. So we found 5% of those trees infected with Prunus necrotic ring spot virus, 1% infected with Prunedorf virus, and that's pretty consistent across the last couple of decades. And this year was the first year that we tested for peach latent mosaic viroid, and we found that in 8% of the trees. Of course, no plumpox virus. This would be a very different presentation if we found that virus. This is another view of those results. I have the block shown across the bottom here, and then the specific viruses detected in each tree. So this reflects the different sizes of blocks that are included in the Budwood program, and the ones with the warmer colors, red, orange, and yellow, are single infections of either virus or the viroid, and then the cooler colors, purple, blue, and green, indicate trees that were infected with 2 of those pathogens in the various combinations. So we do have some sort of problematic cultivars where it's difficult to find virus or viroid negative trees in those blocks, but we continue to work with the growers each year to find better blocks for the Budwood program. I'm not surprised that we found a lot of PLMVD this year because this was the first year that we were testing for it. I did mention briefly that we switched our testing methods from ELISA, which is an antibody-based method. It's not very sensitive, but it's easy to do a lot of samples really quickly. We switched from doing that to PCR-based testing because we're able to include more pathogens more easily, and it actually has ended up being less expensive per sample doing that method. If you're interested in that methods comparison, Brody Cox has a poster on that, so you can check that out. Okay, so I'm going to move on to an update of the Clemson Clean Plant Center, what we've been doing over the last year or two. So this is an overview of our program. So I already talked about the Southeastern Budwood program. That's a main part of the Clemson Clean Plant Center. But we also do diagnostics for more than just the viruses that we listed, right? We're able to do high throughput sequencing, and we're able to test by PCR for a whole list of 26 and growing viruses, viroids, and graft transmissible bacteria. If we are able to detect any viruses, or if we detect any of these pathogens in that material, then it goes into our virus elimination pipeline. And this is the newest part of our program, using a combination of tissue culture and heat therapy to eliminate viruses from those accessions. And then once they are clean, then we can put them in our foundation, which is maintained in a greenhouse at the Musser Farm at Clemson. And these are the sort of elite clean material where we are minimizing the risk of reinfection because it took a lot to get them clean. But this material is available to the public for propagation. So what do y'all think is a clean plant? What words come to mind when I say clean plant? Virus-free? No disease? What was that? True to type, that's a huge issue, yes. What about certified? Anybody think certified? Certified clean plants? So yeah, I would think certified and virus-free are words that probably come to mind. And this might seem like semantics, but I did want to take a minute and go over this because I think it's important, to say what you mean and mean what you say. And really, these plants are not virus-free and they are not certified, at least not in the Southeast. Certification programs are run at the state level. So we have National Clean Plant Center or National Clean Plant Network centers, clean plant centers around the United States that develop clean plants. And we'll discuss what that means in just a second. And then the state departments of agriculture are the ones who actually certify the plants. So me at Clemson, director of the Clemson Clean Plant Center, I don't certify any plants. Certification programs are strictly run by the state departments of agriculture. So it usually works in this way that the clean plants, the foundation, or sometimes referred to as G1 level plants, are maintained at the centers, and then that material is, you can sell either seed or bud sticks to nurseries who will then propagate that under specific conditions specified by the State Department of Ag, and that will be at the G2 level in nursery mother blocks, and then G3 usually indicates increased blocks at nurseries, and then G4 would be the plants that are delivered to the growers, right? Certified clean plants delivered to the growers. Only a few states have certification programs, California, Oregon, and Washington. In the Southeast, we don't have certification programs, which actually I think is kind of a good thing. And the Southeastern Budwood Program trees are negative for 3 viruses and a viroid. So that's sort of our version of a certification program for peach. And certification programs, I think, honestly, they sound really good on paper, and sometimes they cause more problems, especially because we're discovering a lot of new viruses, and we don't know which ones are problematic and which ones aren't. But I do think that it's important that we define what a clean plant is, and I would refer to it as a virus-negative plant, or one that is derived from a virus-negative mother plant. We can't really make the claim that it's virus-free, and so that's why I don't like to use that term, and we don't use certified in the Southeast either. Okay, so the diagnostic process at the Clemson Clean Plant Center targets what we know to be economically harmful or potentially economically harmful pathogens at optimal times of detection, and we test twice before declaring a plant clean. And we test twice by high-throughput sequencing before we are able to put a tree into our greenhouse at the Musser Farm in our foundation. I mentioned this already, but our diagnostics have switched entirely to more sensitive molecular techniques like high-throughput sequencing and PCR-based techniques. That's because they have higher sensitivity, better specificity, and we can test for multiple pathogens at a time. Those are things that you can't do with ELISA testing. So this is the current list of the Prunus panel pathogens. I will say this is what they test for on material that's brought in from overseas. So we don't do any import at the Clemson Clean Plant Center, but our sister centers at Washington State and at UC Davis, they do controlled import, and these are the pathogens that they're looking for. And this list is growing all the time. I did mention this, but I'll say it again, not all of these viruses cause problems in stone fruits, and especially that list gets whittled down even more when you consider just peach. So if we find that an accession is infected with something that we are concerned about, then we can conduct virus elimination therapy. And this is, as I mentioned, the newest part of our program. We are conducting diagnostics and virus elimination currently for 13 accessions. Most of these are off-patent cultivars, but we have Dr. Gassick's Caro-Res and Everez varieties, so they're still going under the diagnostic process, and if they are infected with anything, then we would put them into the virus elimination pipeline. Some of these may not be infected with anything, in which case we just have to test again after a dormancy period, and by high throughput sequencing, and then we can put them straight into Foundation. And our Foundation collection serves as a source of this clean nuclear stock material that's available for propagation. The amount of budwood that you can get from Foundation trees, of course, is small. We generally maintain 2 to 3 trees of each accession in our greenhouse, and these are potted trees, so they're not the same size as normal orchard trees. And each plant is tested by high throughput sequencing twice prior to entry into Foundation. And we also, once we put the trees into Foundation, we don't just let them go after that. Twice a year, we test them for a sub-list of targeted pathogens, ensuring that this collection remains clean. So here's a slide of our current Prunus Foundation located at the Musser Farm. We do have this greenhouse that I showed on greenhouse that we showed on the previous slide. And then we also have a field block. So we have a lot of the same trees, or a lot of the same accessions that are in our greenhouse are also maintained in a field block where we also do maintenance testing every year. And we established the field block basically just to be able to provide more budwood for those accessions if it's needed. So this is a list of cultivars. We have a few peach cultivars, some rootstocks, and 2 plum accessions. And I will take this time to say that if you're interested in receiving material from any of these, or if you're interested in adding cultivars to the Foundation, then please reach out to me either after my talk today or via email is fine too. I like to just kind of compare the Southeastern Budwood Program and Foundation because you can get some things from one that you can't get from the other. The Southeastern Budwood Program has currently 4 participating nurseries and several participating growers every year. These mother trees they're on commercial orchard sites, and they are tested for plum pox virus, Prunus necrotic ring spot, prune dwarf, and now peach latent mosaic viroid. The budwood source trees are, again, we have between 50 and 100 per block, are full-size production trees, and so that provides a maximum amount of budwood that the nurseries are able to collect. And the time to establish a budwood block is just a couple of weeks because, again, these are trees that are maintained by the growers on their properties. So they're doing the hard work for us. The NCPN Foundation is different from the Southeastern Budwood Program in that it is a public program. This is a national program, and so this collection is viewed kind of as part of the whole NCPN fruit tree collection in tandem with the one at Washington State University and the one at UC Davis. Anybody can request material from the NCPN Foundation, and the mother trees in this program in the Foundation are tested for all the known graft transmissible pathogens. So they are like super clean. And these Foundation plants, again, we keep 2 to 3 of the trees indoors, are in pots, and they do provide only limited amounts of budwood. So it's not, we're not going to be able to establish a whole orchard from one of these trees. And making a clean plant can take several years because virus elimination is not for the faint of heart, I have learned. Okay, so I just want to mention some plans for upcoming research and Clean Plant Center activities, and then we can get into discussion after that. So I've just listed here some of our related ongoing research projects. We have a project where for the main viruses and the viroid, we're looking at seasonal titer or seasonal numbers of the virus copies in different tissues of the peach trees over time. And so we're running this study for 2 years. That's Garner Powell up here. That's his study for his master's. And we're doing this so that we can make sure that we are collecting material from the right tissues at the right times for diagnostics, right? We really want to optimize our diagnostics. So that's the point of that project. When we started to find peach latent mosaic viroid everywhere, we realized that this viroid is transmitted on tools. It can be transmitted on grafting tools or even on pruning tools. We know that it is not seed transmitted. We've recently done a small study in Guardian 2 to show that it's not seed transmitted in Guardian, or if it is, it's at a very, very low level and we haven't been able to detect it. So that's good news. But we are testing some solutions for tool sanitation to reduce mechanical transmission of peach latent mosaic viroid. The other one is what I just mentioned about seed transmissibility. We have various studies going on investigating the pollen transmission mechanisms, the role of bees in transmitting these pollen-borne viruses. And the final one that I have mentioned here is our newest project, which is investigating interactions between viruses and the bacterial spot pathogen Xanthomonas species. So this is an emerging story. It's a project that really started in 2020, but we've only really just gotten kind of a little bit of traction on. And this is something that I think as growers you have known for a long time, that viruses, even if you don't see specific virus symptoms in your orchards, you may see that those orchards with viruses are more susceptible to other pathogens, and so that's what we're investigating here with bacterial spot. So again, this is a project where we're focusing on Prunedorf virus and Xanthomonas arboricula pathovar pruni, which causes peach bacterial spot. So some preliminary data on this project was collected by my PhD student. Her name is Daniela, and she went out last summer and she collected leaves from some peach trees that we know are infected with Prunedorf virus and then some virus negative leaves, and she did these detached leaf assays where she spray inoculated them with a suspension of ZAP. And she was then counting the number of bacterial spots that developed on those leaves over time. So what you can see here is that she, this is just the controls. So no bacterial spot developed where she didn't spray it, so that was good. And in purple are the leaves that came from the PDV infected tree, and in blue are the leaves that came from the PDV negative tree. And you can see over time, she's finding more bacterial spots developing on the PDV infected leaves. So we have some other ways of looking at this interaction in orchards and in the greenhouse that we're going to be doing this year. But overall, we definitely have some preliminary data that there seems to be an interaction between this virus and the bacteria. So it underscores the importance of preventing viruses in your orchards. Even if you don't see specific virus symptoms, it can make them more susceptible to other pathogens. Virus prevention is critical because it's really the only time that you have to manage virus diseases, right? Once a tree is infected, it's infected for the rest of its life. And so it can cause, of course, reduced yield, reduced fruit quality, overall weakening, decline of the trees, potential interaction with other pathogens, leading to economic losses for the growers. And management options are extremely limited once the viruses are present in the orchard. That's especially true for the viruses that we encounter in the southeast, Prunus necrotic ring spot and Prunedorf in particular, because they're transmitted through pollen, and you can't kill bees, right? So you can't kill the vectors of these viruses. So we're really limited in management options once the viruses are there. And I'll just finish off by saying that there's recent economic studies across crops that demonstrate the critical nature of clean plant centers and the work to ensure that we are able to produce and provide clean plants to specialty crop industries in the U.S. Okay, so I want to just thank all the peach growers and nurseries who've worked with us over the last few years and continue to work with us, extension agents, the staff at the Clemson Clean Plant Center and in my lab, Dr. Brandon and his lab at UGA, and of course the Clemson Peach team, some of whom are listed here, the Musser Fruit Research Center, both Peach Councils, South Carolina and Georgia, and the National Clean Plant Network. So I think we still have some time for discussion. Okay. Yeah. I don't know who wants to go first. Go ahead.
You saying that the bacterial spot are you saying that it's interacting with these viruses? Because you can do a good copper spray program and you can prevent bacterial spot. Has anybody done any research as far as copper on the viruses?
Not that I know of. And viruses are, they're systemic pathogens. And so you're not, like a foliar spray isn't going to be effective against a virus. We don't know what's going on exactly between viruses and bacteria. I doubt that it's actually a direct interaction between those pathogens. I think it's more likely that the virus is somehow weakening the tree, making it more susceptible to bacterial spot. But we have a few different ideas of what might be going on there. Chalmers, do you have a question? Okay.
The National Clean Plant Network and the Clemson facility, part of the original plan was for that to be able to host low-chill varieties as leaf source. Yet 6 years later, we still don't have any low-chill varieties, and we're not using that center the way it was supposed to be. Because
Yeah.
the processor cannot handle low-chill.
Right.
Can you tell us what's going on there?
As far as us,
Why did that stage never develop where we were going to be the repository for low-chill varieties? Because we don't have a place in the U.S. right now for them.
yeah, I don't know exactly what the low chill varieties are and if any of them are on this list. But this is what we currently have in Foundation. Dr. Scott had a hard time, I think, with working with those centers because he never got the virus elimination part of the program up and running before he retired. I think just the timing didn't work out for him. And so if he wanted to have any variety in Foundation at Clemson, it had to go either to California or to Prosser. Those centers both have a huge backlog of material, and so it's difficult. You can send something there and then you never hear back from them for 10 years. So that is an issue on its own, what has just been getting material back from those centers. Now we have the program started where we can do our own virus elimination here. We're working on it, but it's a slow go. Virus elimination itself, once you even complete the diagnostics and know what the plants are infected with, virus elimination can take several rounds. Some pathogens like Prunus necrotic ring spot virus and peach blight mosaic viroid are extremely difficult to remove by heat therapy. They get into the meristem of the plant, and so it's virus elimination strategies that work against other viruses don't work very well for those. So there's technical challenges on the lab side and just the logistics with all the material having to first go through the other clean plant centers, I think.
Well, you know where I'm going with this. There's a big push that these centers become self-funded.
Right.
And right now, it's all grant-funded.
Right.
And so if we don't have a revenue stream that comes, and the Prosser cannot basically hold anything that's going to child with us. So we can. And so now that you've got the ability to do the heat therapy and everything like that, it's a way of the repository becoming a bank and being able to get fees for that. So
Yeah, that's a good point.
I want you to look at it because we're having to fight very hard to continue to get funding.
Yeah. Yeah, the requests at the NCPN across all crop groups have gone up a lot, and NCPN funding has remained basically flat. So these are the ones that we have in progress right now. So when I say in progress, that means that we've got them into tissue culture, and we're awaiting results from the diagnostics.
Remember red tropic growth prints on this one, and go back to your budwood testing slide where the budwood program.
Yep.
So
I see where you're going.
The growers in the room, I want everybody to understand, because we've worked very hard on this. First of all, explain the ELISA test versus your heat check. You did briefly, but it missed for a number of years a positive detection of one virus that we thought we were testing for and we thought we had a limit.
Yep. Yeah, so The ELISA testing, again, is based on antibodies, and Dr. Scott had been using a batch of antibodies that were generated at the Washington State Center, so that it was generated against the strains of the virus that were prevalent in those orchards in Washington State. And that antibody test was missing a lot of the variants. So when I started, we ran out of that antibody anyway, and I decided, okay, we're just going to purchase the ELISA test from a company, from Agdia. And that worked better, but even that one was missing some variants of Prunus necrotic ring spot virus in this block, in red top. And then we only found that out because we did this comparison test of ELISA on the same samples and testing by PCR on all of those same samples. And I was expecting initially that PCR would be more sensitive, like detecting earlier infections, but less specific, that the antibody-based test would detect more variants, and it actually was the opposite. So the PCR was able to pick up those isolates of the virus that ELISA was missing. And then, of course, there's a lag time with this, right? Because that was only last year that we did that test.
So because of that, we moved mother blocks, we changed a lot of different things. And now we're testing for it, so now we can tell the nurseries to avoid those trees, and I think we can get that cleaned back out. But in the case of gold prints, and many growers in the room here will remember this, somewhere between 2004 and 2007, gold prints completely changed the way it acted. The trees became out weaker, they came out slower, and my belief is that we didn't have a mother block for gold prints, and budwoods were all cut from one orchard in a particular year, and basically we took a virus that we weren't even testing for and changed the variety that everybody has. And so now being able to go back and clean up gold prints and clean up red top, in about 2 years, if she can go through the heat therapy, give us clean wood, we can get a nursery just to plant 100 trees, and we can then turn that into a portion of a block to be the motherwood going forward, and we could have clean trees in probably 3 to 5 years.
Yeah.
But it's going to take that.
Yeah. And our strategy, like combining the efforts of the Budwood program with the foundation, is a really good one, I think. We're kind of uniquely positioned to at least already be sampling from these trees, the ones that we know are at least negative for these pathogens that are difficult to remove by therapy. The other clean plant centers in California and Washington State usually don't have that opportunity, right? They have to deal with whatever gets sent to them that has whatever viruses are in there. But we're able to at least cut off the initial viruses from that list, so that's good news too. But yeah, those are the ones that we've targeted, are the varieties that are included in the Budwood program, so we know that they're already commercially important, and that they have existing problems with viruses and viroids. Yep.
If we have a block of trees that we probably lost 20 to 30% of the trees at 3 years old, if we replant that and the trees had the virus, will that virus be in the soil?
Viruses generally don't survive in the soil, at least not these viruses. And so I wouldn't worry about anything in the soil. I would caution you against any kind of, I don't even know if you do this in peaches, but any kind of top working, because the virus will remain in the rootstock.
But we're going to plant a new tree.
Right, so if you plant a whole new tree, that's fine. My background is in grapevine, and they do a lot of top working in grapevine, so that's where that issue mainly comes in.
Thank you.
Yeah. I will say, too, combining the results of the HTS, the high throughput sequencing survey, with the Budwood program also sheds some good news light on this, because it shows that we don't really have to worry about much other than these viruses that were already on our radar. And so if we can get material cleaned up from these pathogens, and we can remain vigilant, right, do this type of survey every 10 years or so, then I think, we'll be in a good place.
So when you said they were pollen transmitted, or viruses that can be, when we've had these mother blocks that have been established, some of them for 6-8 years, and we'll get an isolated tree that tests positive, we don't see an expansion of that virus in neighboring trees.
Right. Yeah, I should have provided some slides on that. So if you didn't hear what Chalmers said, he said that if we have a budwood block, like let me show the example here. Most of the budwood blocks look like this, where most of the trees are negative for everything, and then you have some isolated positives. And year after year, you know, we continue to include those blocks as budwood blocks in the program until they age out or until they have too many viruses in them. So some of those budwood blocks will go for 6, 7, 8 years, and the viruses don't seem to spread in those blocks. So when you have small inoculum sources like this, pollen transmission is extremely slow, if it happens at all. However, what we did see is one of the blocks that we tested in 2020, again, when we updated the testing method, I think we were able to find more Prunedorf virus than we were finding before. And so we tested one block of summer gold, and it was like 95% infected with Prunedorf virus. And so that's actually the start of the PDV ZAP project. But that budwood block is right across the road from the budwood block of O. Henry. And that orchard, the summer gold orchard that's heavily PDV infected, again, doesn't really show any specific symptoms. You look at that block and it's beautiful. Right across the road, the O. Henry block, we found 4 new PDV infections in 2021, and then another 1 or 2 in 2022, and I forget the 2023 results. But anyway, that's quicker spread than you normally see in a case like this. So I think it's reflective of the initial inoculum source, more so than the actual rate of spread.
Thyroid, I would be concerned. That's pruning.
Right.
So if you see a couple of thyroid positives, that will spread out. I think we've seen that in the early.
Yeah, I've been reading a lot about the viroid lately since we've been finding it, and I don't know the efficiencies of the mechanical transmission. It's very inefficiently also vectored by aphids, but that was just done in like a greenhouse experiment, and their conclusion from that study was that it's probably not, aphid transmission probably isn't really relevant in the field. And so transmission through grafting and transmission through mechanical methods for the viroid are going to be the most critical. There is one study that came out of Italy that showed that the viroid is also transmitted through pollen. And again, I don't know the efficiencies of that, but the mechanical spread would be.
So they prune it and they'll be moving it around.
Right.
They don't feed their pruners after every tree.
Yeah, and we're trying to figure out what you should clean your pruners with to get rid of it, because the things that work for viruses won't necessarily work for viroids. Viroids are extremely stable pathogens, and so you know, if you spray it down with ethanol or something like that, it's not necessarily going to be effective against the viroid. Yeah, question back.
Well, Chris feed on pollen, and they're prolific in the pine trees, and coast pollen are pretty, like, pretty bad, peanut, pretty bad.
Yeah, thrips.
All your early planted crops, fruit, well, spring, we had peaches. So I don't know about aphids, aphids not a real problem southeast, but the real problem is thrips.
Yeah, so thrips have been implicated in spread of the viruses like Prunedorf virus and Prunus necrotic ring spot virus. Those transmission studies are extremely hard to do, especially with thrips, and so the information in the literature is pretty limited as far as their actual contribution to spread. What I think could be happening, and could be also happening with bees, is that those insects sort of facilitate spread of the pollen-borne viruses, where a virus requires a wound to enter a plant in the first place. And so potentially those insects are providing that wound for the virus to get into the plant.
I know they did a study on peanuts where they used different metapropyl, and it ended up fed the virus working. So it's trying to keep off peanuts. I don't know, it's a hard enough to crack.
Yeah, it's like I said, the options for management of the pollen-borne viruses are extremely limited. So trying to reduce the amount of inoculum that we have out there in the first place by, you know, working with the clean plant programs going to be the most efficient method. Yeah, Phil.
Libby, I think it's correct to say that Vegemite will fire is spread by daggers, reported to be in the literature spread by daggers. Now, are any of these viruses, the new viruses, are nematodes that were learned to spread within?
I was expecting to find some nematode-transmitted viruses. There's a couple of them on the list. Where are we here? Here. Sorry, this list is kind of hard to read. I tried to make it pretty, but it made it hard to read. So the, let's see, tomato ring spot and tobacco ring spot virus, Arabis mosaic virus, those are all in the same group, and they are vectored by nematodes, and some of them are also transmitted through pollen. We do some work in blackberries too, and we found tomato ring spot in blackberries. We know that all of these viruses have extremely wide host ranges, so they very often will sort of overwinter in, or survive in weeds, and even in nematodes in the soil. We did not find any of those Nepo viruses at any of the sites. And I was expecting them, but it's good news that we didn't find them. And Dr. Scott said that too, because I remember when I first started here, that was one of the viruses, tomato ring spot in particular, was one that I was wondering about, because I knew that it could be problematic in stone fruit orchards, like in cherry orchards in the Northeast, for example. And he said that he had encountered it once or twice in his entire career. So, our results reflect that as well.
Libby, the new thyroid that you've detected, say its name again, the one we're going to start testing for this year, we haven't been testing.
Peach Late mosaic thyroid that we already have tested for.
But we just started this year.
2023 was the first year that we tested for it, yeah.
And it can be transmitted with prunes.
Yes.
And you found in both your surveys in both states, but in South Carolina, it was a lot more prevalent.
It was pretty prevalent in both states.
So, I mean, we ought to talk to growers about a practice to work with that right now to control it. So we really need to understand, one, what can we use as a dip?
Yep.
Because we're talking about, we can make immediate benefit to our farms if we do this now. But then we also need to understand, what does this thyroid do to the tree? Because again, we haven't tested for it, we have now propagated it.
Yep.
And so it's pretty common, but yet we don't know how bad is it?
We know that it delays bloom. Some of Dr. Riegard's work showed that very well, but it results in decline of the orchards after a few years. Do you want to add something?
Well, there's different variants of it, Chalmers.
Yeah.
When we first did this, back in the 70s when we set up this program, we had it, we didn't know, because we got the material from Procter. We found out that variant never showed any symptoms. So the one I worked with showed a lot of symptoms. That's the one you find in Europe, okay? And this one, after you did sequence it, it's not the same one. So I'm not saying it doesn't do anything, but it seems to have not been a virulent form of this type of thyroid. There are very virulent forms, like she shows some pictures of it. So I'm not discounting it that it's not to be a problem, but or maybe a problem, but it has so far. It hasn't been very obvious. There are very related, the same thyroid that is a problem. It's slightly different. Like there's 4-point mutations different. Dr. Scott looked at it and they sequenced these in Spain. So, but yeah, I was quite surprised. But 30, 40, like 70, whatever that is, 40 some years ago, we had one of these, what they call peach late mosaic thyroid. We never knew. Procter never detected it. And so we went through and we took it out of everything we had in Sandhill Station. We found it did change 1 or 2 varieties when they ripened a little bit. That was the only thing we noticed when we had them cleaned up. Now this is totally, I don't know what the sequence is of this.
Yeah, I have a student this semester who's going to be working on some population genetics, looking at the diversity of the viroid from the samples that we have it from. That's the other advantage of our switch from ELISA to PCR testing is now we have RNA from all of these samples in the freezer that we can keep going back to. With ELISA testing, it's like you process the sample and it's gone. So that's another advantage, is that we're kind of building this library of samples that we can always go back and screen for anything that we want to, really. But yes, as Dr. Riegert said, this is the genome map of peach late mosaic viroid, and the strains that cause this type of symptom here, this, like, they call it, I think it's called the calico strain, has a chunk of 8 bases inserted into the genome that is missing from the rest of the isolates. And I don't think that we have this strain, because I don't know about you, but I haven't really seen anything like that. But the one that we do have potentially could result in decline over time. I know from previous discussions with you about peach late mosaic viroid, because Dr. Riegert was initially looking at it for a method to delay bloom, right? Like to use it for an advantage. But I think you can correct me if I'm wrong, but I think you said that the impacts of it long term causing decline of the tree are kind of why that was abandoned. Yeah.
But the PCR primers you have for this would span that region where the 8 base pair insertion is?
Yeah, viroids are only like between 300 and 400 nucleotides, so you can do a PCR that amplifies the entire genome. So yeah, that's what we'll be doing for looking at these isolates this spring. I think something we can discuss is on adding apple chlorotic leaf spot to the testing. It's not very difficult now to add another pathogen to the testing regime, and I don't know, maybe Elise, you're rolling your eyes because I'm not the one that actually does it. These people are the ones who actually do it. But now that we have, basically the way the process works is like we get the samples in, we extract RNA from the one sample, and then it can be tested for multiple pathogens. Compared to ELISA, where you had to grind and test, grind a sample and test, grind a sample and test. Like you couldn't test for multiple pathogens from the same extract. So now we're able to better add more pathogens. I don't know for sure if apple chlorotic leaf spot is a problem, so I think we could do some follow-up investigations and then decide if we want to add it to the testing regime, but it wouldn't be that hard to do it, so.
So Libby, when you did the orchard surveys, was
Yep.
there any type, you said you went and looked for weak trees, but when you looked at the orchards, was there age associated with that? Did you do 5-year above trees, or, because what I'd love to see, we do this in 5 years from now on 3 to 5-year-old trees, and 90% of this is cleaned up.
Yeah, certainly we could do that. Garner, do you want to address that question?
Some trees were older, but I would say majority were like the 3 to 5 age trees.
Okay.
Generally younger, but some of the orchards were older. So.
For the South Carolina testing, at least. Are we out of time? Okay.
Thank you so much.
Thank you.
All right, next up we have Dr. Chen with USDA. He's a plant breeder at USDA in Byron. Going to give us an update on their breeding program.
Good afternoon, everyone. And I'm so glad we still have a big crowd here as this late afternoons. I think yesterday we have a very good pitch section, and make my talk much easier. You know, our breeders, as you guys, and all kind of pressures, especially in recent years, we suffer so much for the weather. So today, because I only have 15 minutes, of course, I a lot to cover my research. So just to talk about what I'm doing right now in response to this current climate condition for peach productions. I think most growers know that. We don't need to say much about it. Before I start my talk, I probably give an update. We do hire a rootstock breeder. He can come in any time because it has been, since we give the oral offer, now it's already over 4 months. So I think, I don't know, but it's still waiting to the official offers. So he planned to come to the meeting, but he didn't get the offer on time. So I guess probably you guys see him in the future meetings. Anyway, I just changed my title a little bit just to make the logical looks better. So, right now, of course, a breeder have a lot of things to think about. You need your fruit to be marketable. That's, of course, the most important. If your fruit a lot of marketable, there's no need to the growers. Right now, as we say, we have so many trouble related to climate. Especially last year, we lost almost 100% our fruit on trees. So since I was here, I'm 10 years here, actually, it's my 10th year here. I look at the peach productions, they always get a problem with the weather. Either without enough chill, or most cases, spring freeze. So I just see what's going on with this. I think this Pam yesterday used these figures. It's my figures. Anyway, he said I'm from UGA, but it's okay. I'm from Georgia, at least. So everyone knows this chill is a continued decline over the 100 years at Byron. So I think the other location probably the same trends. I want you guys to look at these figures. Think about how to choose your chill for your productions in your areas. That's just a Byron's data, of course. So you can see that's the percentage of each chill ranges. So this anything smaller than 550, that's between 550 and 650. So you can see that's, of course, the average, of course, is here is about 20% is 950 to 1,050. But we talk about your possibility. Don't receive enough chill when you choose the cultivars. So let's just give a table kind of estimate based on the data here. I think that's critical. You think about it when you choose a cultivar in your regions. But the problem is not a big issue for South Carolina. Definitely the issue for the Central Georgia. Let me say that because you guys have a less of a risk of lower chill compared to Middle Georgia and the South Georgia, even the worst in Florida. You heard yesterday to talk about it. So like I say, if you choose a cultivar with 850 at Byron's, you have about every 10 years, you have 1 year you're without a crop. That's almost certain there. So that's just why you put it there, because the breeders need to think about that. If you provide varieties, he plant in ground, every 10 years have 1 year, almost certain you don't have zero fruit on the tree. That's I think a lot of good cultivars. You think about that perspective. Of course, like if you choose like 150, you have almost 20%. It means every 10 years you have 2 years without a crop. So that's just, in my mind, as breeders, you need to think about it for the breeding activities. So that's just about the chill requirement. So that's a newer map just produced by USDA. It's a new plant harvest zone map. You can see yesterday Pam covered very well about the spring freeze. The chill is reduced over the years, but the spring freeze timing coming is still the same. And even lower temperatures. I look at the old map, this temperature dropped about like 1,000 degrees in that neighborhood. So that means you get more extremes freeze temperature in the springs at the same time. There's no delay or get ahead. So that means our cultivar current in use have a way higher risk to suffer the spring freeze like last year's. So that's why I said I still think about it as a breeder think about that. What you can do with this kind of situation? We need a better thing, at least mitigate this kind of risk there. So I just want to give a few words here. You see, all our production in zone 8A and 8B and line A. Line A probably is very limited, but most it is 8B and 8A. South Carolina, North Carolina is all the 8A, and Middle Georgia all the 8Bs. So you can see here, the temperature, the lowest temperature can reach like almost 10 Celsius. None of the peach fruit right now, most of them, and I don't want to none probably is so broad. So that's 2015. I just give you some picture here. It's the 2015 which environs. Most of the tree like this. The fruit are set. That's freezing that happened March 19th, 2015. And the end of the most tree like this, set a lot of buttons without coloring, or some mixed with normal fruit and button with a lot of rotten. And you can see these selections. It's crazy. Every tree button, almost every fruit button, but the color just normal, a size about a quarter of size. But they do have a good one there. So that's the kind of hope, give the breeder hope. Otherwise, the breeder we just kind of cannot do anything for this kind of stuff. So that's 2015. We do have a lot of just like this. Okay, this one is already released. You guys can use right now. I think the Liberty Joy. So that's 2017 already low. I don't want to say too much because that one is because we don't have chew. We only have chew about 250. That picture I took is April 12th. All the trees look like bad. But even that, we still have the lower chill trees. It have a set of fruit pretty good. So it's just give a kind of hope to a breeder to looking for some kind of breeding. So this year more dramatic. So I think in my career, I'll never see my orchard go there. You couldn't find a single fruit, even taste them. So most of my trees like this, you don't see a single fruit on any of these trees. So you guys probably know that, but we do have good ones there. It's set almost a full crop like this one, and this one is a full crop. That's two selection here, and this one is the last selection. And then we did a mid cross on that selection too. So I see that it's a very, very limited number of germplasm in this collection show promise. Let's see that. So other breeders all depend on your genes. Without the genes, you just like in Chinese proverb said, like a wife cannot make a cook without materials, about cook a food without materials. So, but we do have about like a handful of selections. It is a fruit without any problem. So there's probably I'm going to focus more, make more cross with the guy, and probably do more research with that. And that's just a 5 release. I released probably in the past 4 or 5 years. I'm going to start from the latest one, Rich Joy, Liberty Joy, and Crimson Joy. And that's a very recent one, just last year, or this year, last year, or the past year. There's a Cardinal Joy and a May Joy. Anyway, that's a similar fruit character as the most Prince series cultivars. So there's a little bit acid there, and the color very well, yellow flash, and if the late is a probably freestone. So I think you guys see this slide before. Anyway, this one I think is the best of the 5. It's set of fruit almost every year except 2017. This year, let's see, no, 2023, they set about a half crop, not a full crop. Okay. So the problem is okay, I will go further. How many times? Okay, that's just the data show compare with Crystal Heaven and early August because it's almost right in the same neighborhood with those two cultivars. So you can see that generally the size is very similar to among them, and the blushy, this one is much more blushy compared to the other two cultivars, and also have a higher fruit set at Byron's. And this one is the date. So they're a little bit kind of earlier. So that's a Libra Joy. This one's is a kind of I released it because they set up full crop in both year 2015 and 2017. But unfortunately, they didn't set they set out 10 fruit on this tree. 2023. So this still kind of very, very sensitive to like a temperature go really low. So you can see this comparison to the all other 650s. They perform much better compared to the others. So the others just the average is 1.3, but this one is 7, and also 2017 they set almost full crop there too. But just have emphasized that the floor reach never set good cropping in my block anyway. So that's just they never set even closer to my evaluations. Generally, there are about 1,000 fruit there. So that's the Crimson Joy is about 700 chew hours. This one is similar to the Libra Joy, but this one is a little bit higher chew. It's all the crops in the 2015 and 17. That's a part of the motivation we released that. So, but this one is a little bit turned to overcrop would affect size, even a lot of thinnings, especially the weather is good. And sometimes it can develop tip. You already know that if the chew a lot enough, they just turn to prolong the tips. But that's not that problem in Rich Joys. So that's just compared with the similar season, Gila, Harvester, and Red Heaven. They're mostly quite the same, the size, the set. There's no statistical difference, let's say that way. So because my orchard probably manages as good as you guys, you probably see a little bit different from this sense. But they do more attractive, let's say, and a little bit firmly. So that's the two new releases. Early one, very early. This one is 850 chew hours. So I think why release them because in my blog I said the two early ones, Flavor Rich and Car Red, in my 10 years career here is only about 2 or 3 years I have set a full crop. So most years they don't set a crop. But this one, I think they set a crop most year except the 2017. But this one all have a problem. You see here, if the chew is not enough, they can turn to kind of prolonged tips there. And this one is very early. This one is about 2 weeks early than the Car Red. But this one is 650. Okay. This one is just a typical 650 cultivars. They didn't set it well in 2015. I think it's quite sensitive to the freeze. And this year, let's see this year, probably just a 10% crop. It has fruit there, but just very limited numbers. So it just give a warning there. But this one is not released as a kind of a more tolerant to the freeze. But it's early, at Bayer is 2 weeks early in the Car Red. And also have that problem, so it have a kind of tip problem if the chew is not enough. Okay, let's just compare all the 4 cultivar, early cultivars, the May Joy, Color Joy, Flavor Rich, and Car Red. So as I said, the size is quite similar to each other because they're all early, and the set, Car Red, of course, I already said Cardinal because they have higher chews, so the generally set more crop compared to the other 3s. And the others almost kind of look like I have different, but they really look similar. So if you look at the appearance, I think that's my 15th. Is the time? Surface time? Oh, okay. Yeah, any question?
Your Mayjoy, what's your average size on the Mayjoy?
Pardon me?
Average size on the Mayjoy, that early?
Mayjoy probably like 2 inch to 2.5. Probably that's best in my orchard. Let's say that way.
All right, thank you so much. Got Dr. Gesick from Clemson talking about some of the new releases they have coming out.
Okay, thank you. And as Chen said, many of the things were covered yesterday, so that makes it easier for today. And that forced me to do a slight change in what I was going to present, just to kind of answer some of the things that we saw yesterday on effect of the climate influence and some of the diseases. So the title was basically to introduce to you the new series of peaches that are going to be coming out of the Clemson University, and so you can recognize it by the name, and I'll go that later. And overview, basically, I'll just go quickly through what the program goals were or are, and what is the timeline, just to remind you when I started. What is the goal or what is the ideal peach that the program is looking to develop, and then go into the detail on the new releases. So goal, you've seen several of these slides. When I was hired, the goal was to basically combine the fruit quality that already existed with the disease resistance that was missing from the cultivars that we bring from different breeding programs. What we try to do is address several groups of challenges that you heard over the yesterday presentations, mainly climate, then consumer demands for the quality, and actually the quality that they get when they buy the peach in the stores, producer needs, disease tolerance, reduce cost of the producing, and one of the things is retailer education, which I'm now trying to do through naming, and I'll explain that when I go into the why we gave the names, because there are different types of peaches that are fairly different from what is traditionally meant when you say the fresh market peach, but nobody is basically educating consumers or retailers about the differences of these peaches. So going back to the timeline, so remember, I was hired in 2007, started 2008. So starting from basically ground zero. Clemson had a great research going on everything about peach except breeding program. And so that's when I came in. The first crosses were made in 2008, and then first selection in 2011, actually looking in the fruit, and first selection in 2013 when we actually grafted the promising ones and planting them in the advanced selection block. We looked for them in a few years in the advanced selection block, and the first selections, that we call them SC1, 2, and going down the numbers, we sent and distributed to several places, Arkansas, Alabama, and at Clemson, just to look how would they perform, because I was mostly concerned about bacterial spot. At the time, we did not see symptoms at Musser Farm. So I was concerned, okay, maybe my markers are telling me something, but I am actually completely missing the point. And Arkansas peach breeding program is placed in the environment where they every year get hammered by the bacterial spots, so that was the reasoning. So then in 2019, we actually, I got drawn into the rootstock breeding as well, because that's what the problem is, and with the help of the rest of the peach team, we are trying to address some of the things, but my side is genetics and breeding for the replant issues. Basically, we are now in 2024, and the last year we submitted the patenting paperwork for 2 of the cultivars, called them Carores Wonder and Everest Ruby, and I'll go into detail why the names and which ones of those are the SC1, 2, 3, or whatever. And we are preparing the paperwork for the 3rd one. And so that's going to kind of continue now that we are over these 15, 16 years that you actually have to go through when you actually make the first cross until you see the results of your work. And put it outside there and then wait for you guys to yell at me, that's not good or it's good. Okay, so the replicated trials I already hinted about. The first Arkansas and Alabama in the of course, university, either extension programs or breeding programs with my colleagues to look at them. But then recent years, from 2018-19, we established 3 more, one in North Carolina, 2 in the Ridge area in South Carolina, and 1 at Musser Farm. That's a specifically replicated trial with about 10 selections, and we are adding more as they come. And some of the data we'll be showing from those trials. North Carolina trial was supposed to be fruiting last year for the first time. Unfortunately, freeze happened, they did not have any fruit. So we are hoping we'll see the first fruit next year from there. Okay, so now what we were hoping to, as I said, the goal was to combine the fruit quality, and so these were some of the traits that we were looking, and what you told me as an industry meetings that we had, and what I knew that the consumers want, to combine with the disease resistance. The main disease resistances that I was focusing on was bacterial spot, fruit and leaf, and brown rot. For the fruit was actually the higher importance because you cannot sell it if it's damaged. And for the leaf, we just looked at a tree to not be stressed too much and not have a higher than 10 to 20% of defoliation in the summer. Because it's genetics are really more complicated for the leaf, and so it was much simpler to develop the markers to help me eliminate material that's not tolerant to bacterial spot. And then as for the brown rot, the story is not as peachy as for the peach tolerance to bacterial spot, as we will probably never have a resistance, but there are some selections that show promise, and yesterday's talk from Dr. Schnabel's lab on the different methods of treating and rotating chemistry and results that they got while after treating with some of these methods were almost similar to what my selections that I name like tolerant or higher tolerance to brown rot were showing after planted in the orchard that was never sprayed with fungicide. So that's the next topic that I'll have talking to the growers to establish the trials where you will not spray or would have the reduce less numbers or later starting spraying just to test that in your environment. Right now, what we have at Musser Farm, it's next to the Guido's blocks that are also full of brown rot, so we don't have issues with inoculum. It's plenty playing around, and the results were not as bad. The genetics of this are coming from Byron. Dr. Oki long time made a cross and gave me the pollen, and then this is like 4th generation of these crosses because the original tree that carries the tolerance is not really at the fresh market level of quality. Okay, and then of course, climate resilience. I think everybody is like having nightmares with this. Everybody's speaking about that. But how I looked at that, when I was interviewing for the job in 2007, there were no peaches. So that told me, okay, hey, there's something else going on here. And so I immediately starting and focused myself on maybe controlling it to 600 to 800 chill hours. At the time, that was a good range. Now we are looking at the predictions that we need to go even lower. But I didn't look at it as only as a chill because as we heard yesterday from Dr. Shapiro, we have a problem that you bring the low chill material, you get the chilling, they bloom early, and you get the frost, as Chen mentioned, almost at the same time every year, and they got frozen. So it's not just one thing. So the idea I got is, okay, I'll control the chill requirement, but try to increase the heat requirements so I can delay the bloom. So that's the left side of the graph that is showing you how variable chill accumulation is. Recently, we also published a paper with one of the undergraduate students just looking into correlation between the chill portions, as we heard that the chill portion are actually better reflecting what's going on and what you can expect in the season, and trying to give you a quick, dirty method to just, okay, if I know my cultivar needs 350 or 550 chill hours, what does that mean actually in the chill portions? And we'll try to evaluate that through the years, because you need more replicative evaluations than just, we had a historical like 10 years, I think, data to look and compare. On the right-hand side of this was what I started working with Dr. Melgar, because yes, we can have great chilling, great bloom time and everything, but the frost comes and what's then? In 2007, it was April when it hit. That's why you lost everything. So I was thinking, okay, I'm gonna go after a suite of traits I call climate resilience. So control chill, high heat to delay the bloom, and fruitlet freeze tolerance. And so I'll later on show this data for these 3 selections that I'm talking about. So all of that leads into what's the ideal pitch that the program is looking for. So of course, we are trying to expand the ripening season, going earlier so you can hit the markets and get the better prices. When you go early, the size is the issue, the splitting is the issue, and so we are trying to address those things. But the major point is getting everything that we release to have the disease tolerance. Right now, those 3 selections have the tolerance to bacterial spot, but we are still working on the brown rot and making some crosses, and we have one that's bacterial spot tolerant and also high tolerance shows to brown rot. There are some issues with it, but I'll give it to you in the replicated trials, and then you'll tell me if those issues with a slightly around 50% of blush, so not really high blush, but other than that, it looks really nice, and I'll show you that later. So going now to the selections. The cultivar series, why we decided to call them CaroRes and Everest. I was struggling really much because whatever I wanted, somebody already decided to name it that way or call it that series. And so I wanted something that would remind you that it comes from South Carolina and Clemson. That's why I use Caro for the Caro Tiger, Caro King, Caro Red, that were released previously by Clemson University, and wanted that to be reflecting the regular type of softening, like a traditional yellow flesh peach. Res is going for the resistance. Just to remind you that these are the similar, but has that additional benefit of resistance or tolerance to the disease. And the Everest was the way of showing you that these are the peaches that can hang on the tree longer, that are very firm, and can be harvested in the fewer picks, and do not deteriorate in quality if you harvest them in 3 weeks, 3 weeks later than what you originally would do. And those are the ones that require fewer days at the room temperature. After that are melting, juicy, and freestone. So these are the selections that are in replicated trial, but today, and the ripening date against the July print. And then these are the 3 that I will be talking about. The ripening time is the mid-May, late May, and first week of June. And so the numbering was not, the numbering basically was how we selected them in the field, but it did not reflect the ripening time. That's why the earliest one is SC2, then SC9 and SC1, and now you'll see the actual name we gave them. So in this slide, you can see all of the chill requirement and bloom time for all of the selections, and these lines are showing the actually range that I was going for, and this is the line that is the targeted bloom time or later. So of course, hit and miss, which is what Chen also showed, that's what what we are trying to do in the program. So the 3 selections we have, SC2 has a 500 chill requirement, 550, and SC1 is about 800, SC9 about 850. This is one of the things that I wanted to show you, and that actually reflected through the data before we actually got these results. So these graphs are showing you with the dotted line when is the 50% of material we put into the cold environment showing the damage of cold. And the temperatures were starting from 0 to minus 10 Celsius. And so normally how this test is interpreted is giving you that is the temperature. So all of them are similarly at close to -6 or -4.5, but you can see this line going steady flat and then going up. That means that there's no damage almost until -4 in the control conditions, I might say. So this might be a few degrees higher than the -4 in the field, but the order of the cultivars or selections is what we were going after. And that actually reflected in the SC2, or how we call it, Kerores wonder, that we, in the years when we had a late spring frost, we actually had fruit on those. And on these, and even though it lowest chill one, 500 chill hours, and earliest blooming one, but the fruit has a higher tolerance to the freeze. So that gave us kind of hope into the test that we are doing, that we could use it to make difference there and try to incorporate that in breeding. As Chen said, you already saw these slides from several previous talks and PDFs that I uploaded in the different places. But what we tried lately to do is actually to tell you chill portions for them. Because with our climate, that's the only way for you to surely know if you have enough. What's reported when you look at the patenting information is very vague, because you never know if what's calculated, how they calculated the chill, or it's just comparison to others. And so chill portion is what we are now trying to make the push for everybody to report, so that makes your life easier. This is the earlier one, as I said, SC2, and the main take-home message is that fruit tolerance, and that there is a fruit even if there is a scare in the late freezes. The size is fairly good and consistent, excellent taste. Some split appear in some years, but not that much comparing to Caro Red, which is the closest one in the ripening, and it's a high pack-out volume and better taste than some of the comparisons. So showing that as a table, these are the replicated trials from South Carolina and Alabama, because the trees are there older, so we oldest, or the earliest plantings, so we had that data ready. And so you can see the similar time of the harvest, similar mass of the fruit, size, and bricks, and yield. This is basically the first harvest, and so that's the last year we did not I did not put data from the 2023 because the ripening at the Master Farm was out of the whack because of the very early bloom time, and I was worried that it's going to skew my data, so we'll see how we're going to interpret those. Next one is, we call it Kerores Delight. So Kerores is telling you that it's a standard peach with a standard fast melting, and that it's tolerant to bacterial spot. Going back to the SC2, tolerance to bacterial spot was seen in South Carolina and Alabama. Well, in Arkansas, it was hammered. So we are now testing on the with Dr. Hehe tolerance with different isolates to see what's going on there, just to make sure that I'm not telling you one thing and the other thing is happening in the field. And for Kerores Delight, or SC9, go back. Yeah, so these are the characteristics you could have read. It's a higher chill one, 40 to 45 chill portion requires. We also tested the dormancy, cold hardiness of buds during the dormancy with the test with the colleagues in Michigan, just to make sure that maybe we can grow them in the northern climates or colder climates. This one is really beautiful, excellent size, blush, and I'll show you one of the comparisons with the standards, how it fared this year at the Ridge Spring trial. And here is, again, the data. Alabama got those trees later, so the first ripening year was the 2022. And this is thanks to Dr. Elina Koneva. And then these are the information. So very similar bricks, nice size, and ripening. This was in the first week of June, but on our initial selection was the late May, 1st June. So it's like a week after, one after the other. And the SC1 was the first one that we like. That's the first one that has the firm fruit that can hang on the tree, and it's very sweet but not subacid, like let's say Sweet Dream or some of those that are coming out of California. And it's resistant to bacterial spot was confirmed everywhere we had it, including Arkansas, which made me really happy because that's the first one that we trusted the markers and everything, and it did not show any blemishes on the fruit. For all of those, leaves show 10, 15 to 20 maximum damage, and that's confirmed last year in North Carolina as well. And so the next one is, again, comparison and size issue variability. I think in 22 in Alabama, Elina said she had an issue with the late frost, so she didn't have much fruit, and that was that data should have kind of asterisks next to it. And all of these trees, of course, are also Guardian, and you could have read the establishment dates and stuff, or other things that showed in this lower part. And so this is the table that we perform, and thanks to Titan Farm information from the pack out, from the Ridge Spring, you'll see the next one. So we have in the Ridge Spring and near Johnston, and these ripening dates we did not include it just because of the size of the table, but it's about week before in Seneca, and you can see the consistency of the total yield, bricks, firmness when the analysis were made, and the size in grams, in the diameter, bricks, firmness, and all of that for all of them. I'm not sure if this is included, but if you want that information, I can distribute, or we can put it on the website as well for you to have that information. So, so SC9 we did not initially had at the Johnston planted, but now we have it, so hopefully we'll collect the data there as well. And the SC9 is one that we are working on the paperwork right now to submit. And so this is the one that I was referring to as a pack out. So I don't know how well the colors are seen, but basically the highest 3 1/4 and up is the darkest color, and it's only seen in ruby prints and summer prints. And it's right there, this and tiny line this. And then goes the 2 3/4 and 3. But what's interesting here is that the Carol's Delight, 90% of fruit are 2 3/4 and 3/4, and out of this, like 40% are 3 1/4 and higher, and the rest is the 3 inches size. And this is Everest Ruby. The line is right here, so about 50% of fruit is in the 3-inch size. And so this is really a nice comparison, and thanks to Titan Farm for collecting that data for us. Carol S. Wonder is also doing well, but the majority of fruit are from 2.5 and higher. some even proportion of 10% in the 3 inches. And, and this is, of course, Carrot, Summer prints and Ruby prints, so you can see the comparison. This is the slide that I added, and I'm probably over the time because of those talks yesterday. So when student group talked about doing these biologicals with chemistries and planting and about disease severity index, I remember that the few selections in our non-sprayed plot behaved similarly. So what you see here is the material collected from that orchard brought into the lab, and the progression of brown rot looked after 3, 5, and 7 days. And these are the percentage of the fruit showing infection. And so and this is how they look from the order from left to right. And this is the one, and these days are against July prints as well. And so this is the one I was telling you that looks really nice for the fresh market peach. Slightly, I mean, the coloration is not really fully red or more red, 75% like I would like it, but this one is the 25, this one, and this one is this one here. So they're really nice, and then this is like next year, how the fruit looks of this one. So this is something now for us to look at, because we completely did not spray it at the farm, but there are potentially chance to do it, maybe delay spray or reduce spray to see what's going on and what can we do. Of course, that's this one. Of course, none of it will be possible without funding and your support whenever we need to reach for the funding. And the people, I'm just standing here talking, but many people are behind me and working with me, collecting data, taking care of the orchards, including my lab members, Massive Fruit Research Farm crew, Clemson University Peach team, and all other colleagues that we talk and communicate with. So that's about it. And since I'm fairly over the time, I think we can continue into that discussion where I can answer the question if there is a question. Yeah, go ahead, Dario. Yeah. That's the thing. So the question was, I was talking about the slow softening type of a cultivar, and am I going to change the protocol from a harvest or suggest that to the growers? That's exactly what I'm trying to do, and that is what we tested with this particular Everest Ruby. What we did, we harvested it when the growers would harvest it, and then we went harvested week after week after week after. So month after the commercial harvest, they were still firm, no degradation in quality. We put each harvest into the post-harvest test, 2 weeks in the cold, and there was no internal damage or any deterioration in the quality. Because that was one of the big unknowns that nobody knew. Yes, I'll delay the harvest and they'll taste maybe sweeter and be better, but what if it develops the post-harvest issues, because you delay the harvest? So that's one of the big things, especially because the labor is an issue. So if you can just condense it and harvest it 3 weeks later than you would initially want to, because they are firm, they are big size, they are completely red, so it's really hard to determine when to harvest. And even 5-6 weeks after the commercial harvest, maybe 80% of fruit on the tree were still firm, and some started shriveling a bit. That's kind of the characteristics.
Is that a non-melting flesh?
No, melting. So after a few days at the room temperature, it's the freestone, melting, juicy, everything you want in the peach. But if you bite it when you pick it from the tree, it's like an apple. It's crispy and crunchy like an apple.
Some people don't like that.
Yeah. I wanted to toss this one away, and then my colleague said, don't do that. My wife likes it to cut it in the salad. So I sent it to him, and while it was transporting across US, it got those few days, because I didn't want to pay, of course, the highest price of shipping. And then he said, well, this is not that firm. I said, oh, wait a minute, how it's not that firm? And then we actually got the sample again and tested it, and everybody really likes it. I mean, that's the one I ship everywhere when everybody wants to taste what are the different types. Okay, so I think in the program, sorry, I'm taking your job away from you. It said that it would be the discussion with growers in what is it next that we, Chen and I in this case, need to look for or breed for. And as you see the timeline, it takes us 15 years to get anything to you, so the sooner we anticipate something, the better chances that we're going to have some material that will work for what you need. So it seems that we answered all of the questions. Good. What I wanted to say, after the yesterday's talks and everything, it looked like, oh my, doom and gloom, you know, the climate resilience, this changing everything. Two of us, we are trying to make the change, and we are looking and finding the variability. We have lots of trees, so when something happens, there's always a silver lining, and there's always a few of those that are cropping, but nothing else is, and stuff like that. So it's the hardest thing is actually testing it in different environments, because we never know what's going to show up. Yeah. Thank you. And you are absolutely correct, because for years people were focusing on the chilling, and you know, if you grow something in like Michigan, they don't care because they have plenty of chilling, but then spring frost is still an issue for them. And so that's that number of heat units that the tree needs. And we are in like a problematic zone, because at the same time they can accumulate chill and heat. So, go ahead. Oh, that's $1 million question. Dr. Oki published the paper and showed that it's really tricky and really hard. So what we tried to do, we took the historical data of weather data, bloom time, and then we know the chilling requirement, but we also did the forcing just to make sure that those chilling requirements are correct. And then we calculated the heat requirement, but from the moment each individual cultivar actually got satisfied chill requirement. Because usually what people do, they have like a cutoff date, and from that date, which is not correct, because, yeah.
But I know it satisfies chill.
Yeah. I mean, that's what happened to us last year. And because this year we have a nice and steady cold weather, which is different than last year when we had the spikes of hot weather, which was, I think yesterday Jose talked a bit about that, and the climatologist professor talked about that those extremes are what triggers plants. If they got their chill satisfied, or at least 50% satisfied, then they're accumulating together chill and heat. And that's what tricks them to bloom earlier. And then, of course, what I noticed throughout the years, the late spring frost danger is around Easter. So it has something to do with the moon phases and whatnot, but that's, when we survive that, we are good. And then you know how the Easter goes, sometimes March, sometimes April, and then we never know when it's going to hit. But problem is if they bloom like last year in the mid of February, the whole farm, and Clemson University was in full bloom, then basically when the late March freeze came, where we didn't have wind machines, there was it. And we had a crop on Master Farm thanks to the wind machines. That's, I need to say that. Yeah, go ahead, Dario. That's an excellent question. What Dario was making a comment. It's all nice to work on the chill requirement and high heat requirement, but if you push bloom too late, the temperatures are very hot during the bloom and fruit set, and then affects the fruit set. And so how do we how do we account for that? Right now, we did not have many years of that issue at Clemson Station, but we did have it last year, that during the bloom, the temperatures were really high, and so that's a valid question that we need to look into. And I mean, it's more problematic for lower chill regions like South Georgia and Florida, but unfortunately, as the weather patterns shown, it's probably coming up in our region as well. So we need to address that. What I'm looking at many of the things in breeding comes by chance. You know, you do your best, but sometimes you find your selection that has what you wanted even without going for it. And so that's what happened with my SE2. KRS Wonder, because I did not know the chill. Yes, my goal was the 600 to 800. It has 500. So I said, oh, it's slightly lower. I'm gonna. Then it bloomed early. Oh shoot! Now I'm done. You know, blooms early, low chill, and then that's the only one that had fruit. You know, so it is a combination of things. And one of the things that nobody talks about, and I think Jose just touched it for shortly, is the fruit development period. So how many days or growing degree hours or whatever the tree needs from the moment it sets the fruit till the moment you can gonna harvest it. And so that's some other things that we should also look into. You know, there's no shortage of problems, so but there is another way to go. You can go to almond. Almond. Almond has some of the traits that we want in peach, and Tom Graziel made the crosses and had the selections, and we are trying to incorporate that to break some of these ties between the traits. So, and almond, yes, and the Spanish breeding programs have the advanced selections from almond peach crosses that they isolated regions of the genome to target different traits. So it's not really a bad quality of fruit. They did lots of back crossing and everything. So we don't necessarily need to go to China because it's really hard, not hard, impossible to get anything out of there, you know, and any collaboration. So then we can go to Europe, you know, and get collaboration from there.
Did you release any germ plasmid or copper?
Sorry?
Did you release any germ plasmid?
No, what they did it mostly for molecular studies, but they did the back crossing to re-establish the peach with 1 or 2 traits from almond. And so they have that RIL whole orchard available, and if you want it, they'll give it to you. The problem is plumpox virus bringing it here, and what Libby was talking about, you know, how many years it would okay. That's why I never tried. But there is, but pollen, I mean, we could try something. So.
All right. Thank you. Thank you for all of our speakers. Next, we have Ms. Kay Rensel with the National Peach Council.
Okay. Thank you so much. Thank you so much for giving me time on the program here this afternoon. I promised someone, and I can't remember exactly who it was, that I wouldn't take a lot of time, but I will share the most important things with you because I understand I'm between you and football games. That's what someone told me. And so I haven't personally checked, but again, I am Kay Rensel, Executive Director for the National Peach Council, and what I try to do for you every year is give you an update on what we've been working on your behalf, and then also we just have to have a very brief business meeting as is required by our bylaws. So let me just kind of get started here real quick. One of the most important things that we've been working on in the last 2 years has been the addition of fresh peach purchases to the USDA purchasing programs. And without doing a deep dive into all of the detail, there are 5 different purchasing programs that USDA does on behalf of the recipients. One of them, probably the largest one that you will recognize, is the school lunch and breakfast program. But in addition to that, there's other programs. There's something called TFAP, and that's for emergencies. So anytime that there's a disaster and, they send out the various arms of the government to help with that. There's also emergency food that goes along with that for the residents in the affected area, as well as the volunteers that are there. And there's the Indian and reservation tribal reservation programs. So those are just probably the three biggest ones. But then they also do one for the food banks, excuse me. And that's where a lot of the purchases that we've been working on the last 2 years for the FRESH program have gone. And you can see that in 2023, last year, USDA, now they just give me a combined number, so I can't necessarily split all of them out very easily, but last year, they purchased nearly 60 million pounds of peaches. That's both frozen and fresh. And it had a value of over $100 million. So the industry was truly a direct benefactor from that. Part of the reason that we went to USDA in the first place was we understood the number of trees that had been planted across the country, the production capacity that was coming into play, and also the world dynamics and the ability to be able to market all of those peaches. Let me just step back and say one of the major benefits for that is it put peaches in the hands and the mouths of people who otherwise may not have been able to afford them. For example, in the school lunch program, a lot of them are still the 4.4 ounce frozen cups of peaches, but through the FRESH program and in areas of the country where schools are in play during the purchasing period, we put fresh peaches there as well. So it certainly has been a benefit, and just kind of a small spin when David said yesterday how the next generation is asking for some of these fruits, specifically peaches and the various berries that are out there. I think part of that has to do with the way all of us in the specialty crop industry have learned to work with USDA and open these doors for those one-day consumers that are out there. If you are interested in being a supplier and being a part of the USDA purchasing programs on fresh or frozen peaches, I encourage you to begin the process. It takes a while. It is rather cumbersome, but you would have time if you are interested. But understand that you personally don't have to be one of the people selling to USDA. You will still benefit, because by the fact that we've been able to move those fresh peaches out of what was strictly a retail and a food service marketplace has now opened the door and lifted the floor of the market in the last 2 years. In 2023, we requested some specification modifications on the bids themselves. We added the volume fill boxes and in addition to the 2-layer and the 2-pound bags. This year, we're also going to ask for another slight modification, and that would be adding an additional size peach that can be put in the bags. What happened last year is without peaches in the Southeast, it forced the majority of the sales to be provided by the West Coast suppliers, and in the end, they had very large fruit, so the size that was to go in the bag was really a struggle. So for all of you that, and there's probably everybody in this room, I am praying that you all have a great crop this year, which means hopefully when we go to USDA this year, they're going to increase their purchase. At least that will be our specific request to them. So I'm very excited to tell you we already have in place. We're going to, actually, I'll be in discussion next week with USDA on preliminary conversations about the volumes that we would be looking at. And it's going to be well over the dollar volume that they had in 2023. So we'll keep you posted along the way. We'll certainly give you the good news as we get it. I can tell you, as a messenger in my conversations with USDA, they are absolutely thrilled with the nation's peach industry. We have consistently delivered quality. They have had no quality issues as far as, and they tell me when there are rejected loads. There was not a single rejected load anywhere in the country of all of those peaches that were shipped in 2023. So congratulations, but please keep up the good work, because as soon as there's problems, I'm the first person who knows, you will know as well, and that dramatically will impact their ability to be able to justify purchases moving forward. So that's the good news. And as I learned, that's one of the things that has been of most value to the industry. So let me just kind of share some of the other things that I've worked on and continue to work on for you. Farm Bill. I happen to be from the state that the chairman of the House Ag Committee is located in, Glenn Thompson. I have had years of relationship with him, and so I went to him ahead of time and I said, so, what do we need to do? He said, we're going to get this thing passed. It's going to happen by August of 2023. Conversation over the holidays and throughout the fall was, he's trying. They have it pretty well drafted. They just now have to wait for their space on the House floor to bring it to the floor. There will be cuts. I don't know where all they're going to be. I have testified twice in listening sessions on behalf of the industry on the value of protect and preserve crop insurance. Specialty crops are vital to America's farmers as well as American consumers. All of those key points that are important to having you stay in business. So know that I'm working on it, but I don't control Washington any more than you guys do, but it certainly is an interesting time to be there. One of the other things that we continue to provide for you is the annual statistical yearbook. It was published in late July, early August, and I do apologize because this, from what I understand, could be even more valuable to you, but our hands are tied by the timing of the information that comes from USDA, and that's what that entire report is based upon. So when we published the report in 2023, it only took you through the 2022 crop. So we're a little behind. You can see the trends. If you want a copy of it, please reach out to me and let me know if you haven't seen it or downloaded it. It is out there, it is available, and we'd be happy to share it with you. With regard to some of the other work, I do spend a fair amount of time in Washington, D.C., and it was primarily because of the Farm Bill and some of the other issues that we work on. Participating in the meetings at USDA with various segments and arms of USDA that we need to keep apprised of what's going on in our industry. But we also continue to make sure that they are aware of the issues that are ongoing with labor and immigration. As someone in the industry stated earlier today, aside from some of these various issues that you're having with production, labor is one of your biggest concerns and biggest problems. There's been lots of marker bills, marker letters placed and sent on behalf of agriculture, but right now we're in a standstill situation. We are trying to make sure that the AWARE kind of is held at a flat level, but I make no promises. I work in coalition with a lot of the other smaller commodities, and they just don't understand that you're not adversely affecting the local workers anymore. So the need for an adverse wage rate is not really a valid statement. So we'll keep at it. It's a tough one, but that's part of what I'm here to do for you. Aside from the industry relations, we try to get out a few times a year. I can't obviously get to everybody, so anytime you're having a state or a regional meeting and you'd like me to just, I don't need to say anything, just to be there to visit, I'm happy to do that. Would love to visit your operations as time permits. Also, you have heard probably a day and a half of the value and the input of what all of the research projects are doing for our industry. And so when a researcher is working on a proposal, a letter, a lot of times needs to be sent in support. How important is this research project to the industry? And I am happy to do that to all of you in the room. If you don't know that already, please just reach out to me. I'm happy to support on behalf of the industry. One of the other capacities that I sometimes serve in, I'm serving this year, but not in the tree fruit or stone fruit area, is an industry relevancy reviewer. So when all of those grants are submitted, even if I do have a letter of support there, that is not considered a conflict of interest. It actually provides strength to, in some cases, educate the other people that are not from this industry and assigned to it to review the actual proposals. So I'm happy to do it, and please, when someone says to you, whether you're a producer, whatever your role is in the industry, would you provide a letter of support? That is one of the biggest things and most weighted things that we look at as reviewers. What does the rest of the industry say? How important is it to them? So I encourage you to please support everybody that's working on our behalf. We continue to track all of the supply line issues. They kind of fluctuate up and down. I'm not sure if it's a good thing or a bad thing. All of the freighters that I saw up and down the river the last 2.5 days, it is good because goods are flowing in. I didn't see quite as many going out, but you know, trade is important to us, and so are those supply lines. And there's going to be some disruptions, I'm sure, just based on global dynamics right now, but that's a whole other topic that we can talk about, and we'll do that over football. And finally, I wanted to let you know that the U.S. Council did sponsor David yesterday. If you felt that that presentation was of value, please let us know. If you felt it was not of value, please let us know. We were trying to find a different perspective in presenting to you, the industry, the global dynamics of the peach industry and the opportunities that may or may not be out there. So if you, again, I'd like to know either way. It'll help us moving forward. We're happy to sponsor those types of speakers for you if it's something that you feel is beneficial. And then I'm just going to do a little deeper dive into this FRESH program that we did. This kind of just shows you the trends there of where we've been. You could see from the day I walked in the door of working for the Peach Council, the frozen people, the frozen inside of the industry had already been doing these bonus buy purchases for years. It was how they helped to alleviate pressure on the fresh market. But now we can do both, and that makes it even stronger. So that's why you see that frozen goes all the way back to 2019. Yes, I've been with you a little longer than that, but you can see that on the frozen side across all of the programs, they purchased over 35 million pounds of peaches last year, and then on the fresh side it was 24.4. So it's those are good numbers for our second year, and almost, well, just slightly shy of double there from one year to the next. But again, that has to do with the commitment and the performance that this industry put forward to deliver good quality. And then I just thought I'd show you the number side of it. These are actually the dollars that USDA spends. When we go to USDA, we ask for a dollar amount in a purchase. And I want to remind any of you that participate in this is not low-ball pricing, okay? It is fair market pricing. So we have begun to establish ourselves now with USDA from the standpoint that if we keep delivering and the recipients are still looking for peaches, both fresh and frozen, USDA could be the largest purchaser of peaches annually across the board, larger than any other retailer out there. So the opportunity is real, the opportunity is good for us. So thank you for what you've done to deliver excellent quality.
Hey, how much did that come outside of California, though?
In 2023, a lot of it did come from California. There were small buys in the Mid-Atlantic region, but again, it had to do with volume. So one thing I will say is if you bid, you have to deliver. You have to deliver, even if you have to go buy it from somebody else. And I know that that happened to somebody, but they understood that if they did not deliver, then they went on this list that is one that you don't want to be on because it takes you a while to get off that list of I bid but didn't supply. So, but the year before, when we were at the, in 2022, that came from all across the country. All across the country that year, Southeast, California, Mid-Atlantic. I don't think there were any from, I have to think, I don't think there were any from Michigan. So.
When does the bid have to be in a particular year?
Well, it just depends, Greg. Right now, USDA has told us they need 3 months to approve our request, okay? So that's why we're going to be placing our request by the end of January, so that hopefully, cross my fingers, cross my toes, everything else, we're going to know by the end of April what they're going to put out on bid. Then they need another month to get all of their systems in place and surveys done. Then they'll announce what the bids are.
So these would be past frost by May, so.
Yeah, we try to work within the parameters of the industry and all of the red tape in Washington. So, but we are grateful that they did add fresh. And might I also say one more thing? We were the first fresh fruit or vegetable to be purchased through this bonus buy program. So that's pretty exciting. And as I said, they couldn't be more thrilled. So, any other questions? Okay, we'll get to the really easy part now. So last year, as we did, and we'll do right now, is we'll have a mini annual meeting in terms of the business meeting part of it. These were the minutes from last year. It's basically a call to order. And I'm looking for, hold on, I'm looking for a clock. Oh, it's not on my screen. Got it. Okay. These were the minutes that we were from last year's meeting. They were approved from the year before, and the annual, as was the annual financial report. It was presented to all of you. Your board of directors last year was approved, and that's pretty much what we did from a business standpoint, but we will also allow you the opportunity to discuss things. So are there any objections to the approval of last year's minutes? Okay, thank you. Moving on then, just so you can see a 2-year presentation here of the balance sheet of the National Peach Council. We were down slightly in 2023. We are respectful and understand that sometimes when you don't have a crop, it makes it a little bit harder to continue to support the organization. But I will tell you that we're going to be sending out our annual membership reminders in the next 2 weeks, so please consider it. Please put us up there as something that is of value to you and your organization as we continue to work and move forward on behalf of the peach industry. Although it is down some, this is still considered a very good financial picture for us as an organization. And we don't spend a lot. We just get a lot out of the little bit that we do spend, and that's always been my philosophy. I know how hard you all work, so I work equally as hard trying to conserve the resources. Okay, then moving on to the board of directors. This year, there is a rotation, and Jason Rogers, who was serving as your first vice president, has agreed to move up to president, and I do believe, I know he's not here, and I apologize if I spelled his name wrong, so I have to go back and check that for sure. The first vice president will be determined once we approve the rest of the board, as will the treasurer. For those of you that are long-term members of and supporters of the National Peach Council, you may or may not have known that Gary Van Sickle from California was serving as our treasurer. He has served as the treasurer for, my goodness, at least 10 or 12 years. And he did tell me, I'm allowed to tell you, he is officially retiring in July of 2024. So he will be stepping aside, and then among the board members, they can decide who will step in there. And just as a matter of information, it's a very easy thing with electronic communications now. Everything works very smoothly. So that means that Sean will move to our immediate past president, and then our at-large board members, two of which will move up and assume roles as vice president and treasurer, would be Huff Childs from Virginia, Jeff Hopkins from South Carolina, Casey Jones from California, Tom Beaver from New Jersey. Yes, we were able to get representation out of New Jersey. Mike Thurlow from California. You may or may not know him. He is with Mountain View Sales, the organization out there, and then also Chris Yance. So if, now I need to take my notes now. If I could have a motion to approve that? Someone? Anyone? Okay. Jeff? Second? Okay. All those in favor? Oh, I'm sorry. Any other nominations from the floor? I guess I do have to ask that. I kind of did it out of order, sorry. Nope. Come on, don't everyone jump up and say, I want to. Okay. All those in favor? Opposed? Okay. Well, I look forward to working with all of these people and all of you throughout the year. I just want to say it is my pleasure to continue working for you. I certainly enjoy what I do. And my phone line is always on. The only reason I wouldn't answer it is if I'm tied up doing something else and not available, but I reassure you, I will get back to you. If there's an issue that you want to talk about privately, it stays privately. If there's anything that I can do to help you and help the industry, issues, even at dinner last night, I became aware of a different issue now that we need to talk about and work on behalf of the industry. So that's what I'm here to do. I'm here to work for you and also improve your ability to do business and do it profitably. So with that, thank you very much. I'm here, and you got my phone number, my email, and everything else. So, yes, ma'am.
Yeah, I want to just say thank you for all the support you have and last-minute letters and one of those the planes that will be barred on your vacations and everything, but
It's okay.
Thank you.
Oh, my pleasure. And yes, it was, but it's okay. She helped me out. It was last minute, but she drafted it, and it was all good. So I appreciate that extra effort on your side as well. So, okay. Well, thank you. Appreciate everybody, and I'm going to continue to pray for a good year for all of us.
All right. Thank you so much.